Once Mendel had finished his monohybrid crosses, that is, crossing two different heterozygotes with each other and seeing what happened, he became curious about how combinations of traits would be inherited. He wanted to do dihybrid crosses, that is, two plants that are hybridized for two different characteristics. How would their offspring appear? So, to give you a clear example of that, the color of peas could be yellow or green. Yellow is dominant, green is recessive. The shape of the peas could be wrinkled or round. Here are some wrinkled peas right here. Here are some round peas right here. In this case, wrinkled is the recessive form and round is the dominant form. For color, yellow is the dominant form and green, which I'll give a lowercase y, is the recessive form. Remember, for the same trait, use the same letter just in different capitalizations. So you don't do y and g for yellow and green, you do capital Y and lowercase y. He wanted to know, in the F2 generation, if the plant received yellow peas, would it also necessarily have round peas? If it got green peas, would they necessarily be wrinkled peas? Does it inherit all of the characteristics of one parent, or will we see every possible combination? These two possibilities have different names. In dependent assortment, alleles for two traits are always inherited together. So if you start out with yellow round peas, all of the offspring that have yellow peas also have round peas. In independent assortment, the alleles appear in all possible combinations. So you would have yellow round peas, green wrinkled peas, yellow wrinkled peas, and green round peas. So which would it be? Well, if you think about it in those terms, in the pea generation, we've created true breeding plants for two different traits. Yellow and round, green and wrinkled. Now, these parent plants, because they only have one kind of allele for each gamete, and they're going to make, their sperm and egg cells here, they can each only produce one kind of gamete, as far as these traits are concerned. This one can only create recessive wrinkled, recessive green gametes. This one can only create dominant round, dominant yellow gametes. And if you mix them together in the F1 generation, obviously the dominant traits are going to show through. So you'll see nothing but round yellow peas. Now that's true in both situations. Right? That part doesn't change. It's in the F2 generation that things start to be a little bit different. If roundness and yellowness are always inherited together, we could combine those two concepts and call the trait round yellowness, right? And then you would only see round yellow peas and green wrinkled peas. So it would be just a normal 3-to-1 phenotypic ratio here. You would get, it would be no different than if we did a monohybrid cross. 3 round yellows to 1 green wrinkled. But that's not what he ended up seeing. Instead, what he saw was every possible combination, but not in equal amounts. So, this F1 generation plant, it could create gametes that had two dominant alleles, round and yellow. It could create ones that had wrinkled yellow alleles, round green alleles, or wrinkled green alleles. And then same thing with the second plant down here. It could create all 4 of those possible combinations, which ends up giving us a 16 square Punnett square. Now, for the most part, the dominant traits are still going to win out in most of these combinations. It's only where you get 2 copies of a recessive allele that you end up with recessive traits. So, the bulk of these plants are going to have 2 dominant sets of traits, 9 out of 16 yellow round peas. We'll get 3 of each type of dominant recessive combination. So 3 recessive green but dominant round, 3 recessive wrinkled but dominant yellow, and then just one way where we can get nothing but recessive alleles, so you end up with green wrinkled peas. This is a normal ratio for a dihybrid cross. This is a 9 to 3 to 3 to 1 phenotypic ratio. Okay. So, a dihybrid cross occurs when we are creating hybrids for 2 different traits. That is, in the F1 generation, this organism is heterozygous for 2 different traits. The offspring that we create in the F2 generation display all possible combinations of traits, but they do it in a 9 to 3 to 3 to 1 phenotypic ratio. This also applies to other organisms. So again, I'll use my own genetics as an example. My mother has blue eyes and blonde hair. My father has brown eyes and brown hair. I have blue eyes and brown hair. So inheriting one set of traits from a parent didn't mean that I necessarily would inherit all of their traits. I have a nice mix of different traits. So Mendel's dihybrid cross experiments supported the hypothesis that each pair of alleles segregates independently of the other pairs during gamete formation. Thus, the inheritance of one character has no effect on the inheritance of another. You can get all possible combinations, which should be obvious to anyone who's ever purchased peas in the grocery store because green round peas are the ones we tend to buy. This is called the Law of Independent Assortment. It doesn't matter if you inherit this trait from a parent, it doesn't mean you'll get all of their traits. Now, Mendel didn't figure out all of the laws of genetics, all of the rules, because there are actually some situations where if you inherit one gene from a parent, you are more likely to inherit other genes from them. Specifically, if you have 2 genes that are riding on the same chromosome, then chances are if you get one of the genes from that parent, you'll get all of the other ones that are attached to that same chromosome. But because of crossing over, it's not a 100% guarantee. Sometimes genes will become unlinked from each other. In general, though, the law of independent assortment does hold true because most of your genes will be on different chromosomes. So, here are all the different chromosomes that you have. Each one has potentially thousands of different genes located on it. Now, we have mapped out the human genome fairly well, and we, for example, know that if you go to the 11th chromosome, that is the 11th largest chromosome, the largest one is given number 1, and then the second largest is number 2, and so on and so forth. You will find at a particular location the gene for insulin. There are many different versions of that gene, some of them a little bit more functional than others, and this can influence your predisposition to a condition, for example, like diabetes, as insulin is a peptide hormone that helps control your reuptake of blood sugar. Now, if you have different genes riding on this same chromosome, then if you inherit one version of the insulin gene, you'll probably inherit versions of other genes that are on this same chromosome. But again, not a guarantee. This should be a familiar looking image. This is a recap of meiosis, but I've taken the chromosomes and I've labeled them with which alleles you find on each one. So again, every organism has 2 sets of homologous chromosomes. For each pair of chromosomes, you have 2 copies. Your 2 copies can have different alleles on them, different versions of that same gene. So over here on the left, we have the P with its 2 sets of chromosomes, both of which have the dominant yellow and the dominant round alleles on them. Over here, we have 2 sets of homologous chromosomes with the wrinkled and green alleles, all recessive here. Now, the F1 generation, obviously, is going to have all heterozygous traits. They're all going to have one of each kind of allele. But when these organisms create their gametes, it's going to have a bunch of different possible combinations. You can either have round yellow alleles, you can have wrinkled green alleles, you can have wrinkled yellow alleles, or you can have round green alleles. This is true for the other parent as well, and the combination of the combination of gametes from these two parents is what accounts for the 9:3 to 3:1 ratio. If you add up all of these numbers, right, if you just add these, you'll find that there are 16 different possibilities. We have 4 different kinds of sperm or egg cells that are possible. 4 times 4 gets you your 16 different possibilities. So that's where this comes from. Humans also have dominant and recessive traits. So freckles, for example, as it turns out, that is a dominant trait. Now, one thing you might notice is that most people do not have an overabundance of freckles on their face. And that is because dominant traits aren't necessarily the most common trait in the population. How common a trait is, and whether it's dominant or recessive, have really nothing to do with each other. There are some traits that are exceedingly rare in the population, but as it turns out, if you have them, it is a dominant trait, so you will exhibit that. Dominant doesn't mean that the trait is better. It doesn't mean that it's likely an advantage. It doesn't mean that it's more common. It doesn't mean anything like that. The only thing it means is that if you have 2 different copies of the allele, the dominant one is the one that determines the phenotype. The widow's peak, which is this little point of hair on the hairline here, is dominant over having a straight hairline. The shape of your earlobes is also genetically determined by a single trait. Whether you have free earlobes, which is what I have here, which is where the lobe kind of hangs down almost like a pendulum before it attaches to the side of your face, versus attached earlobes, which is the recessive trait, where the earlobe attaches basically as soon as it can to the side here, and you don't have that little flap hanging down here. We're going to use earlobes in order to investigate another kind of genetics problem that we could talk about. So this is called a pedigree, a family pedigree. Basically, it's a family tree, but it's a family tree where we've color-coded it with a bit of genetic information. Here, we have squares and circles. Squares are male. Circles are female. Okay, so all the circles you see, these are women in the family. All the squares you see, these are males in the family. Individuals that are colored in have the recessive phenotype. Individuals that are open shapes, white-colored shapes, have the dominant phenotype. Using just that information, we can figure out what the genotypes of almost every single person in this family tree is. And this is how. So whenever you're doing any kind of genetics problem, the first thing you have to do is write down the information that you absolutely know at the beginning. These people with the colored in shapes, here, 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 so on, you know that they have the recessive phenotype. That tells you something immediately about their genotype. If you have the recessive phenotype, you must have the homozygous recessive genotype. The only way you can have the recessive phenotype is if you have 2 copies of the recessive allele. So right away, we filled in half of the information, half the genotypes on this family tree with just that one bit of surface level information. Similarly, we can fill in 50% of the information that's remaining on this chart just with another piece of information. If you have the dominant phenotype, you must have at least one copy of the dominant allele. So everyone that's circled, everyone that has a white shape here, we can fill in half their genetic information. Now, there's still another half to fill in. And some people will throw up their arms and say, well, that's all the information that's available to us. We don't know what that other allele is. But in any genetics problem, after you've finished filling in the information that's just on the surface, next you look between generations, and that will give you some clues. Here are your two big clues for these two individuals. So let's take a look at these parents in the P generation. They had offspring with a mixture of traits, including two with two recessive alleles. These alleles had to come from someplace. So that means that this parent had a recessive allele to give. This parent had a recessive allele to give. They can't have two recessive alleles in the F1 generation unless both of these parents had a recessive allele to give. So right away, we can fill in any parent that has homozygous recessive offspring. They must have at least one recessive allele. Same thing here. We have a homozygous recessive offspring. We have two parents that we don't have their information filled in, but if they both gave their offspring a recessive allele, they must have a recessive allele to give. So that fills in neatly right there. The last two remaining individuals here are here and here. And unfortunately, they don't have any offspring for us to go from. So maybe we can figure something out from their parents. Well, we know that they got one dominant allele from one parent, but they could have gotten either of these other two alleles from the other one. Same thing up here. This individual got one allele, one dominant allele from one of these parents, and they could have gotten any other allele from the other, and we wouldn't know just from looking at them. So those individuals are actually still mysteries. We don't know. We don't know what those last two are. Still, though, just from using a little bit of logic and what we learned about the basic rules of genetics from Gregor Mendel, we were able to fill out the majority of information inside this chart with only just a little bit left over. If you do not know what something's genotype is, you can write in what you do know, followed by a blank for the mystery allele there. However, I would caution you against jumping to the conclusion that you don't have enough information to work something out. Usually, I will give you enough information to solve the problem. So do try to logic your way through these things. Some genetic disorders that are passed from generation to generation turn out to be recessive disorders. So you might have parents who are carriers for a certain disorder. That means that they have this recessive gene, but because they also have a dominant gene, they don't have the disorder themselves. In this case, we're looking at a form of congenital hearing loss, inherited deafness. Both these parents are carriers for this gene, but they themselves don't display these characteristics. However, if they were to have kids, they go to a genetic counselor, they know that hearing loss is common in both of their families, they want to know how likely is it that any kid that we have is going to be born deaf. Well, you would do a normal Punnett square for this. Both of these parents are going to have 50% of their gametes with a dominant allele, 50% with a recessive allele. This means that there's a 25% chance that they'll create offspring that is fully able to hear and also not a carrier for this condition. There is a 50% chance that they're going to create offspring that are carriers for this condition but can hear just fine, just like the parents. But there is, in fact, a 25% chance that they're going to create offspring that are born deaf as a result.